look, not maybe the last time, but the last look, exercise 4, some anatomy and physiology tissue samples for you folks to look at, pretty much the same as you've done, and then we'll move on from there as far as the microscope is concerned. Okay, so Chapter 7, we're talking about the cell membrane. We mentioned last time about its structure, the lipid bilayer, so that's the basic thing we want to keep in mind. We talked about the fluid mosaic model proposed by Singer and Nicholson, 1973. Why the model is basically correct in saying that cell membranes are fluid and they are made up of a mosaic, a combination of different substances. And I tried to give you some analogies to help us to better understand that. Questions, comments, the beginning of Chapter 7. Okay, let's continue again for about 30 minutes or so. I'm expecting someone to come in and talk to me, so at that point you all can start with your lab work. But anyway, we'll see.
Page 125, do we need to know what amphipathic?
Yes. You need to know the term. We've introduced it before, amphipathic. We say that the cell membrane is amphipathic. What's that mean? That means that the phospholipid, it's better probably to say that the phospholipid molecules that make up the cell membrane are amphipathic, wouldn't it? So, meaning that the phospholipid tails are highly hydrophobic, the heads are highly hydrophilic. So yeah, I would say that's a good term to remember. Okay, other questions? Okay, the cell membrane is a mosaic of materials, components. One of the components that we commonly find within cell membranes are membrane proteins. In other words, proteins that are either attached to or embedded in some way in the bilipid membrane, in the double lipid membrane. And on page 128, there's a list of 6 different kinds of proteins that we might find in or on the membrane. Let's talk about some general terms. First of all, in general things. Again, proteins in this book are highlighted in purple. Proteins are highlighted in purple, just like carbohydrates are colored green. So that's consistent. So here you see on page 128 a number of these different globular-shaped proteins attached to or embedded in the membrane. Proteins that span the entire membrane and stick out, top and bottom, are called transmembrane proteins, Trans, because they transit the entire membrane, membrane proteins, transmembrane proteins. So, in fact, looks like almost all of these would fall into that category. Okay, now 6 different kinds of proteins have been identified that are associated with the cell membrane. But there's one that's very important to us, and it happens to be the first one listed at the top of 128, transport proteins. So we'll spend some time with those. Let's look at the other ones, because we can quickly take care of them. Some of the proteins associated with these membranes function as enzymes. So we call them enzymes, and they catalyze biochemical reactions in place as they are attached to or span the cell membrane. And this can happen on the cell membrane itself. It can happen on any of the internal membranes as well. Notice that he's shown 2 of them, and often they occur in groups or in clusters together, because one enzyme is not enough to complete the biochemical reaction from start to finish. So here in this case, the triangle's converted to a square, the square is converted to a sphere, and so on and so forth, completing whatever biochemical reaction is going on there. So that's one other type of protein that's associated with membranes. We'll just call them enzymes. 3, signal transduction proteins are also found associated with membranes. What these do is to receive chemical messages by molecules called signaling molecules that by virtue of their shape attach themselves to the signal transduction protein. They call it a receptor here. And then something happens that defines really what transduction is. What is transduction? If something is transduced, what's that mean?
Smaller.
Not necessarily smaller, but a change in form as far as the signal is concerned is affected. And typically, chemical signals are converted to electrical signals within the cell when this process happens, as an example. So signal transduction typically means that a chemical message brought to the receptor by virtue of the shape of the receptor and the signaling molecule is converted many times to an electrical message that stimulates the cell to do something. So these kinds of proteins are found on cell membranes as well. 4, some proteins are there just to be able to recognize other cells. So cell-to-cell recognition is affected, like we said before, through the ECM, that extracellular matrix, which includes or can include a number of proteins that recognize one another by virtue of their shape. Some proteins, 1, 2, 3, 4, 5th kind of protein, I guess, are used to join, attach one cell to another, kind of like structural building blocks, if you will. And we just call this intercellular joining. The proteins just fit together and help hold two adjacent cells together. And then certainly there are a number of these proteins, number 6, that function to be part or serve as part of the ECM we talked about earlier, identifying, tagging, naming, or labeling the cell to which they are attached. Okay, but transport is the most important function of these kinds of proteins that we are interested in. So let's take another look at transport proteins and stick with the figure at the top. There are several different ways that transport proteins can do their jobs. And what is their job? Their job, obviously, is to allow for the transport, for the movement of materials across the cell membrane, now listen carefully, which otherwise could not move across the cell membrane on their own. Okay, I'll repeat that. A basic function of transport proteins, these proteins embedded in cell membranes, is to facilitate, to help, to aid in the movement of materials which otherwise could not move through or across the cell membrane in or out of the cell by themselves on their own. Okay, so what's that saying? That brings up a real important idea. What's the important idea? Not all substances can easily move across cell membranes. Some of those substances are needed or required by the cell. So how is the cell to obtain them or get rid of them? That's where transport proteins come in. That's where transport proteins come in. So, and I'll come back to this in just a minute. There are a number of substances. Think about substances that are large, that just can't pass through the cell membrane. Some of those substances can pass through the cell membrane with the help of transport proteins. Think about substances that are highly polar, charged. If the inside of the cell membrane is hydrophobic, they are not going to have an easy time passing through that cell membrane because they will be repulsed just like water is. In fact, water, and we'll get to this in just a second, finds it difficult to pass through any cell membrane. You might find that surprising. Water's not a very big molecule. We know the cell requires water, but under normal circumstances, without a special kind of transport protein, water molecules cannot and will not pass through a typical cell membrane. Why? Tell me why, so I can make sure you understand it. Why? Because water molecules cannot easily pass through a cell membrane because the inside of the cell membrane, where the two tails, where the two fatty acid chains are pointing at one another, those two tails are highly hydrophobic. They're going to repel that water. It's not going to be easy to get a water molecule through a cell membrane. And if water molecules can't pass through cell membranes, and water molecules are polar, they have positive and negative sides to them, then that means that other things that are polar or charged cannot easily pass through the cell membrane either. Things like sodium ions that the cell requires, potassium ions that the cell requires, magnesium that the cell requires, or any polar substance or charged substance for that matter. So, the importance, therefore, of a transport protein being there to allow for the passage of those materials in and out of the cell is underscored, isn't it? Were it not for these transport proteins, the cell couldn't even take up or get rid of water. Okay, so that's kind of a general overview. If you look real carefully at your drawing, you'll actually see that there are several different kinds, two different kinds in this picture, of transport proteins. On the left, you got to look real carefully here, is a transport protein that possesses a hydrophilic channel through which materials like water and other charged substances can pass because they're attracted to it. The hydrophilic channel replaces the hydrophobic normal environment of the interior of the lipid bilayer, doesn't it? Yeah. So here's a ready-made channel for water, polar molecules, ions, other charged substances to move in and out of the cell through the membrane. And in fact, we call this a channel protein. It's a channel protein. And that's typically how it works. It's a channel protein, C-H-A-N-N-E-L, protein. One of the examples of a channel protein, back to water, and I'm not sure where it's mentioned here in the book, maybe it is, maybe it's not. Oh yeah, here it is, bottom of page 130, is the aquaporin. Turn the page over for a minute, and let's use this as an example to highlight channel proteins. An aquaporin, as the name implies, is a channel protein that allows for the passage of water, whereas otherwise water would not normally pass. Notice some of the characteristics of aquaporins. Each aquaporin, each one of these little transport protein and its channel, allows entry of up to 3 billion water molecules per second, passing in a row, single file, through its channel. And apparently there are 10 lanes, because then it says which fits 10 at a time. Without aquaporins, a small part of these water molecules only would pass through the cell membrane. So that's a good example of a channel protein, and that's what allows water to move in and out of the cell. We haven't talked about the mechanism that controls that. We'll get to that later. We're just talking about the transport protein here. Okay, so a few terms. Transport protein, channel protein, aquaporin. Now, go back to your page 128, top, the transport protein picture. On the right-hand side of that little picture is a different kind of transport protein. It's a different kind of transport protein. What happens here is that something, like a molecule or a substance, attaches itself to the protein, and in so doing, changes the shape of that protein. The protein is very malleable. It's very subject to a little bit of outside influence. So when this material, whatever it may be, changes the shape of the protein thrusts or propels, if you will, that material across the membrane. That can happen with or without the aid of energy in the form of ATP. That can happen with or without the aid of energy in the form of ATP. We call this kind of protein a carrier protein. We call this form of protein a carrier protein because essentially it's carrying whatever needs to get across the membrane with it. Okay, so that's just a general class of a different kind of transport protein, the carrier protein. And again, carrier proteins can work with or without energy. And again, that depends on the mechanism driving the entire process that we haven't talked about. Okay, so there's another way to move materials across cell membranes that maybe would have a hard time moving across those same cell membranes. Questions or comments on that? Because we'll build on that. Now let's talk about the cell membrane in general, again keeping the role of transport proteins in mind. What are some of the things that can and cannot naturally pass through cell membranes? Well, the book has a nice little paragraph. I'll highlight it in my book on page 130, and it's the paragraph titled, it's one single paragraph, The Permeability of the Lipid Bilayer. So in my book, I've kind of highlighted it like that. Okay, but it's a nice little reference. So let me paraphrase it for you. You want to know what things normally would go through or move across cell membranes and what things would not. All right, let's see what it says. Nonpolar substances, like CO2, that's an important substance, isn't it? Cells have to get rid of CO2. It's a waste product. Oxygen, oh, that's important. Cells need that. They are hydrophobic. They can therefore dissolve in the lipid bilayer and cross it easily. No transport proteins required. Good for them. So the cell has no inherent problem exchanging oxygen or CO2. Why? They're nonpolar. No charge associated with them. They're not going to be rejected by the hydrophobic interior of the cell membrane, right? No problem. Plus, they're small. Okay. However, oh, there's that word again. However, direct passage of a number of substances is stopped due to the hydrophobic nature of the interior of the membrane. What do they list? Polar molecules such as glucose and other sugars have a problem. Not only because they're a little bit bigger, but also because they are somewhat polar. When they get into the inside of the membrane, they're going to be rejected. They're going to be repelled, just like water would be. Water, mentioned as well. A charged atom or molecule. What do we call charged atoms or molecules? Ions. Whether they're positive or negative, it doesn't matter. They're even less likely to go through the cell membrane. Not only are they charged and repelled, but if they're in water, if they're dissolved in water, they're surrounded by a hydration shell. Water's polar, more charges, more problems. They're going to be repelled as well. Okay, so, and then that's where those transport proteins come in and play a role in terms of aiding or facilitating or helping these hard-to-pass substances pass through the membrane. I mentioned sodium ions, potassium ions, calcium ions, magnesium ions, chloride, Cl- ions as well. Those do not pass through the cell membrane very easily. Okay, so the point is, do you see why transport proteins are important? And they're part of the mosaic model of Singer and Nicholson that describes in full what the cell membrane looks like. So now we know why they're there. Now we know why they are there as well. So I think they're probably the most important kind of protein as far as we're concerned in terms of understanding that permeability of the cell membrane itself and whether that's going to work or not. Okay, questions about that? I really want you to understand that because, again, we'll kind of build on that. Questions about that, the permeability of the bilayer. In general, you should know what substances can and cannot easily pass on their own through the cell membrane and what substances are going to need some help in the form typically of a certain kind of transport protein to do so. If I made up a fictitious substance and told you its characteristics, you could probably tell me if it would or would not pass through the cell membrane, right? Yeah. Just based on, is it real big? What kind of charge does it have? Polar, non-polar, that kind of thing. Okay, questions about this? All right. Now, what have we not talked about as far as the consideration of the movement of materials across cell membranes? We haven't talked about the force, the mechanism. Remember, I keep telling you mechanism is important when you describe something. It's not good enough just to describe it, to observe it, but you need to attempt to explain how it works, what makes it go, what drives it. To answer that question, what would you say in the case of most instances of cell biology and the consideration of the movement across cell membranes would be the primary driving force? Diffusion is the answer. Diffusion. Diffusion. Let me define diffusion in several different ways because I think you're all kind of intuitively aware of it and know what it means, and you've certainly seen examples of that in everyday life. Let's start with this definition. Diffusion is or involves the movement of a substance from an area or region where it is highly concentrated to an area or region where it is of a lesser concentration. Or to even simplify it further, diffusion is the movement of a substance from a place where there is more of it to a place where there is less of it. That's pretty simple, isn't it? That's about the simplest definition of diffusion that I can offer you. And intuitively, that makes sense to most of us. Everybody got that? Was any energy required to do that? No. Diffusion, physicists say, is a passive physical process. When we use the word passive, no energy is required. Diffusion is a passive physical process. If I were to go to this corner of the room and open up a bottle of perfume and basically keep the room closed, the students in the other corner of the room would eventually, over time, detect that perfume, right? And then we would say, what? That perfume diffused, in this case, through the air. Now that's a medium, isn't it? until some of the molecules from that perfume were detected in the other corner of the room. That's an example of diffusion. So to add to our definition a little bit, diffusion can work through a medium. It can be air, it could be water, it could be just about anything. Sharks are able to detect molecules of blood over long distances through salt water and fresh water too. We could say that that blood diffused from whatever source it was to the shark or until it was detected by the shark. So diffusion can occur through a substance or through a medium. It doesn't have to occur across a cell membrane. It can, but it doesn't have to. Let's add to our definition a little bit more. When we talk about the movement of a material from an area where it is of a relatively high concentration to one where the concentration is relatively low, we say that the material moves down its own concentration gradient. So we'll make it a little more technical here. The material moves down its own concentration gradient. And again, that just means that the material moves from high to low concentration. Whether that's across the room, through water, through wood, or across a cell membrane, it doesn't matter. That's still diffusion. Does that definition make sense? Okay, so those terms are used when we describe diffusion. In lab, page 131, some of you may have done an experiment whereby you take a beaker and separate water in the beaker by means of an artificial membrane or something that's permeable, put a colored dye in one side of the beaker, and then time or watch or observe the diffusion of that dye through or across that barrier, membrane, whatever it may be, to the other side of the beaker. But what's the end result of that kind of experiment? An equilibrium is reached, isn't it? Whereby there's a balance in terms of the amount of dye on either side of that, whatever that may be, barrier. And that's what happens in nature as well. What if you put two different substances in the same environment? We find that the diffusion of one does not affect the diffusion of the other. The two do not interact. They don't interfere with one another. And you can show that with two different kinds of dyes or colors of dyes until that magical state we call equilibrium is reached. When equilibrium is reached, that doesn't mean that there's nothing going on. It just means that the relative concentrations on either side of the membrane are about the same. Okay, so diffusion, this passive physical process, is one of the major, if not the major, driving forces that accounts for the movement of materials across cell membranes, whether that's into a cell or out of a cell. So, could we not now predict, knowing some information about substances and their concentrations inside and outside of cells, which direction they would move? And what would eventually happen over time? Yes. When we do that all the time. Cell biologists make predictions about the movements of materials into and out of cells when cells are placed in different environments all of the time. And that's very important. Okay, at this point, we'll stop our lecture because my contact is here. What I want you to do, and you're all familiar with this, same lab exercise. There are 8 sample slides for you folks to look at. They're listed on the board. Make sure when you take them out and look at them that you're looking at the right thing. There are some variations. And if you have questions, I will be here to help you. Make your drawings, make your photographs, make them good.